Assalamu alaikum. Welcome to the study world with Mushu. Today I am taking a lecture on a molecular biology. That is the subject of the MSc Zoology, Part 1 and Semester 2. Today's topic is genome organization. In this video, we are going to discuss about what is genome, what is C-value, what is C-value paradox, what is Scott curve. Let's study. When we're talking about a genome is a nothing but a complete set of a DNA that contains a genetic materials in an organisms. Any genetic materials that stored in a DNA that is nothing but a genome. When C value, the term C value refers to the amount of total DNA present in the haploid nucleus. In a single nucleus, in a haploid nucleus, the amount of a total amount of a DNA present is nothing but a C value of that organism, and they look the structure of their organisms. In simply, we can also define as the linear relationship between the genome size and the complexity of organism. If the genome size is smaller, the organism will be a simpler, and if the genome size is a larger, the complexity of an organism is a increased. We can say in easy term, the complexity of an organism is a directly proportional to the genome size, but it's not true. Why? We see in next slide. To prove that the C-value paradox. The term is given by a C. A. Thomas in 1971 when repeated sequences of the DNA were discovered, explaining in every case no relationship between genome size and complexity of organism. The DNA content of an organism genome is related to the morphological complexity of eukaryotes, but it is observed different in higher eukaryotes. In higher eukaryotes, there is no correlation between complexity and a genomic size. This is called a C-value paradox. Genome size is the total amount of DNA contained within the copy of single genome. For example, salamander have 40 times more DNA in comparison to humans, where the human are a more complex organisms compared to the salamander. When we're talking about a Scott curve, it is a technique for measuring the complexity and the size of DNA or a genome. The technique was developed by Roy Britton and Eric Davidson in 1960. The technique is based on the principle of DNA renaturation kinetics. The rate of renaturation is directly proportional to the number of times the sequences are present in the genome. Given enough time, all DNA that is denatured will dissociate or reannihilate in a given DNA sample. The more repetitive sequence, the less will be the time taken for renaturation. The process involves denaturation of DNA by heating and allowed to reannihilate by cooling. The renaturation of DNA is assessed and stereoscopically large DNA. In next video, we will discuss about repetitive and non-repetitive sequences in a genome organization in details.